everyone, my name is Sia Sharma and today I'm going to be telling you about the various organelles present in a cell. So first, let's do a quick recap of what we know till now. We know that a cell has many parts in it called organelles, which carry out many functions in the cell. In my previous video, I discussed about two organelles. The nucleus, which is also known as the brain of the cell, and this is because it contains the genetic material or DNA of the cell. The nucleus also has the nucleolus, which makes a thing called ribosomes. The second organelle I discussed was the mitochondria. The mitochondria is also known as the powerhouse of the cell, and it basically converts energy from the food that we eat. This energy is then stored in the form of ATP molecules, which are then used by the cell. So this is about the nucleus and the mitochondria. Now we're going to be looking at a few more of the cell organelles. So first, let's start with the endoplasmic reticulum. The endoplasmic reticulum is of two parts: the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, or SER. The reason it's called the rough endoplasmic reticulum is because it has these things called ribosomes attached on its surface, which makes the surface rough. On the smooth endoplasmic reticulum, there are no ribosomes attached, and so the surface is smooth. The basic function of these ribosomes is that they make proteins, which is then used by the cell and carry out most of the functions of the cell. And since most of the ribosomes are attached to the rough endoplasmic reticulum, we can say that the rough endoplasmic reticulum synthesizes proteins. The smooth endoplasmic reticulum, it synthesizes lipids, which are found in the cell membrane and also have various other functions. The smooth endoplasmic reticulum also helps in the detoxification of the cell. Both the rough endoplasmic reticulum and the smooth endoplasmic reticulum are made of a thing called cisteine. So cisteine are the series of flattened stacked pouches. So if you can see over here, you can see like this series of flattened stacked pouches. So this structure is just called cisteine. Over here we can see a cell, and this part that you can see over here is the rough endoplasmic reticulum. If you notice, the rough endoplasmic reticulum is also connected to the nuclear envelope, and it surrounds the nucleus. A bit farther away from the nucleus, we can see the smooth endoplasmic reticulum. So now if you look at the rough endoplasmic reticulum, we know that it synthesizes proteins. But where do the proteins go later? So in the rough endoplasmic reticulum, these proteins, they bud off from the rough endoplasmic reticulum and go into another organelle called the Golgi apparatus, which we will look at in a second. So these proteins, they bud off from the endoplasmic reticulum in vesicles. Vesicles are basically just the tiny sacs that transport materials inside and outside the cell. So over here we can see this is how a vesicle looks like, and we can see the proteins inside the vesicle. So these proteins, because they bud off of the endoplasmic reticulum, so this vesicle over here is also made of the endoplasmic reticulum itself. So over here you can see the microscopic image of the endoplasmic reticulums. This is the rough ER, and these dots that you can see over here, those are the ribosomes, and this is the smooth ER. So next, now let's come to the Golgi apparatus. So after the proteins, they come from the endoplasmic reticulum, they go into the Golgi apparatus. The Golgi apparatus is also known as the post office of the cell. And this is because its function is to process and package all the proteins and the lipids. So over here, we can see in this diagram, this part that you can see over here, that is the Golgi apparatus. And we can see a vesicle budding off containing all the proteins coming to the Golgi apparatus. This Golgi apparatus will then process and package all of these proteins. And after they're processed and packaged, it will again bud off from the Golgi apparatus into another vesicle. This vesicle is then sent to its specific destination from the Golgi, wherever it's needed. So let's say if suppose this vesicle was to be sent, or the proteins inside the vesicle were to be sent outside the cell, then the vesicle would merge with the cell membrane and the proteins would be expelled, as you can see in this GIF over here. So now, over here, let's suppose this is another protein. The Golgi apparatus, it kind of puts a tag on the protein, telling it where to go. So for this protein over here, it's destined to be sent to the membrane of the cell. So it gets transported there inside a vesicle. Over here, you can see the microscopic image of the Golgi apparatus. Now finally, coming to the lysosomes. You can see the lysosomes over here in a cell, and this is kind of what they look like. The lysosomes are also called the trash can of the cells, and this is because they break down all of the excess and all of the damaged cell parts. So over here, let's take an example that a mitochondria is there in the cell, and for some reason it gets damaged and it's not working anymore. So what the lysosomes will do is they will break down this mitochondria into the basic structural units. These basic structural units are then sent back into the cytoplasm and they can be reused in the cell to make other things in the cell. So now how these lysosomes actually break down all these things? They have a specific enzyme inside them called the acid hydrolase. These acid hydrolases are formed in the Golgi apparatus itself, and they bud off from the Golgi apparatus into a specific vesicle, which is the lysosome. These acid hydrolases, they only function in an acidic environment. So inside of the lysosome, it's very acidic. It has a pH of around 4 to 5, and so that's why these acid hydrolases, they can function properly inside the lysosome. Also, one more thing that is there, that proteins which are formed in the Golgi apparatus, they are packaged and processed in the Golgi apparatus, they are also sometimes destined for these lysosomes. Over here, we can see the image of a lysosome in a microscope. So this is all that I wanted to see today. This is actually the end of cell and all of the cell parts. So in my future videos, I'll probably be discussing about the central dogma of molecular biology. But that's it for today. Thank you very much. Please stay safe, stay healthy. Thank you.